For the introduction. Hello everyone, my name is Sina Dadgar, second year of PhD student in the University of Arkansas. Today I'm planning to talk about characterizing reoxygenation kinetics in head and neck tumor xenografts using diffuse reflectance spectroscopy. So to get it started, each year 600,000 new patients are diagnosed with head and neck squamous cell carcinoma. And unlike other cancers, patients from head and neck cell carcinoma are diagnosed in a late stage, such that when these patients show up into clinic, about 65% of them already show locally advanced disease and immediately begin chemotherapy in combination with radiation therapy. Now, the biggest challenge facing these patients is treatment failure because of locoregional recurrence, meaning that tumor showed resistance to the given treatment. And currently methods which are being used to evaluate how well a treatment is working on tumor is limited to anatomical measurements of tumor volume or imaging modalities such as FDG uptake using PET scan, which takes place after 6 to 8 weeks after initial treatment. So indeed, there is a critical need to identify this resistance in the first week of treatment to avoid toxic side effect of chemotherapy and radiation therapy. Now, if we take a closer look to how radiation works, is that exposure of cells to ionizing radiation creates free radicals either in the form of DNA or water molecule. So in the presence of oxygen, this free radical in DNA will be forming peroxy radical, which is capable of modifying DNA. However, in the absence of oxygen, this free radical will be reduced to its original form, and the cell eventually will be allowed to repair itself. That's why patients with hypoxic tumors tend to be more resistant to the treatment and survive less, whereas patients with considerable amount of oxygen in their tumors tend to be sensitive to the treatment. Nowadays, the way radiation works, at least for head and neck cancer patients, is that radiation is divided to several dose fractions, and the rationale behind that is a tumor microenvironment consists of cells with different levels of oxygen. So the radiation kills some of the oxygenated cells that I explained in the previous slide. Again, this is presumed, and after death of oxygenated cells, the oxygen they were consuming is now available to more hypoxic cells, and iteration of this dose fractionation kills more and more cells. But the challenge is right now we don't have a mechanism, we don't have a tool to measure whether this reoxygenation is actually taking place or not. And that is the whole motivation of our project, is that can we use optical measurements to monitor whether this reoxygenation is actually taking place or not. So what we are suggesting for this study is the technology of diffuse reflectance spectroscopy, which is a non-invasive method which is capable of recognizing underlying pathology of the tissue. So the idea is to illuminate the tissue with an optical fiber probe. This visible light will be interacting with the tissue. Some part of it will be absorbed by strong absorbers such as oxygenated and deoxygenated hemoglobin. And some part of it will be scattered by light scatterers such as cell nuclei and collagen. And eventually, some portion of the light will be reflected out of the tissue. So using empirical or analytical models, we can analyze this reflected light to extract meaningful functional information out of it. So here you can see representative diffuse reflectance spectra with different scattering and absorption values. So our goal with this study is to evaluate whether diffuse reflectance spectroscopy is capable of measuring if reoxygenation is happening between doses or not. Specifically, we wanted to first validate the sensitivity of diffuse reflectance spectroscopy with already established methods, and second, to differentiate the reoxygenation kinetics in radiation-sensitive and radiation-resistant models of cancer. So for our first goal, we used an inverse lookup table-based model to characterize the oxygenation, the oxygen saturation that diffuse reflectance spectroscopy is picking up. So the distance between source and detected that we used in our probe is 2.25 millimeters, and we measured the sampling depth of that to be 1.8 millimeters. So to validate this, we used pimonidazole, which is a very accepted hypoxia marker, and we ensured to compare pimonidazole from the same depth that the sampling depth of our tumor is. So to test all of this, we injected 2 different head and neck cancer cell lines to 20 nude mice. They didn't receive any treatment whatsoever. We allowed them to grow until they hit the volume of 1,500 millimeter cube. And then we injected pimonidazole intraperitoneally. After 55 minutes, we conducted diffuse reflectance spectroscopy. And immediately after that, we excise the tumor, snap freeze it, and save it for immunohistochemistry. So here, we can see the diffuse reflectance spectra of 2 representative animals with illustrating different levels of oxygen. We can clearly see that the sample with high level of oxygen has 2 pronounced alpha and beta bands, whereas the poorly oxygenated tumor has a single dip. And using our model, we quantified the absorption spectrum corresponding to these samples. Again, we saw the same thing, that well-oxygenated tumor has 2 bands and poorly oxygenated has only 1. Now, to validate these measurements, we looked at their hypoxia level, and interestingly, we found out that the tumor with high level of oxygen had low level of hypoxic fraction, whereas tumor with low level of oxygen had high level of hypoxic fraction. These pictures, by the way, these images represent the pimonidazole, where binarized images, white pixels, represent PIMO-positive areas. Now, this is only for 2 animals, 2 samples. When we look at our entire data set, we see that oxygen saturation is negatively correlated with hypoxic fraction. This is indicating that hypoxia is formed when microvasculature is not capable of meeting the demand from surrounding tissue. When we look at hemoglobin and oxygenated hemoglobin, we saw the same concordance. And interestingly, very interestingly, we saw that scarring is positively correlated with hypoxic fraction. Now, scientists from Duke University have previously proved that scarring is positively correlated with necrotic cells. So what we are seeing here could mean that highly hypoxic tissues, highly hypoxic tumors, also have some number of necrotic cells. So for our second objective, we injected a human head and neck cancer cell line to 48 animals. We allowed them to grow until they reached a volume of 200 millimeter cube. And we designated that day to be day 1. Starting that day, by the way, after day 1, we randomly distributed these animals to control and radiation group. Starting from day 1, for a period of 14 days, we conducted daily optical spectroscopic measurements, and after day 14, we monitored how the volume of tumor changed. Whereas for radiation group, the schedule was pretty much the same, except the fact that we radiated these animals with 2 rays of radiation twice weekly for 2 weeks. basically on days 1, 4, 7, and 10. So the first evident observation that we made was that these two groups have very different growth patterns, such that the group that was radiated had much slower growth compared to control group. And given the fact that we radiated these animals only 4 times with 2 rays, this is clearly indicating that this cell line, USCC22B, to be a sensitive cell line to radiation. Now, if you look at the reflectance of reflectance of a tumor belonging to an animal before and after radiation, we see that post-radiation spectra in blue diamonds has, the dips that it has is more pronounced when you compare it to pre-radiation, which is illustrated in red colors. So when we use our lookup table model to extract absorption spectrum, we clearly see the same thing where the oxygen saturation belonging to blue spectrum is higher than red spectrum. One other thing that we noticed is that the intensity of these two spectrum are not changing in R. This is an indicator of scattering not changing. And indeed, when we look at scattering, it was not really changing. So now that we have absorption and scattering, we can further break these two to its components. We model scattering to have a negative power law dependence, whereas absorption is a linear sum of absorbing chromophores inside the tissue. And from scattering, negative power law dependence of scattering, we see a significant increase from dose 1 to dose 4 over time, whereas scattering power was decreasing. However, we didn't see any significance here. As far as oxygenation goes, we observed that after each dose, immediately oxygen saturation increased, but after 24 hours and 48 hours, it decreased. So in average, we saw that radiation-induced reoxygenation is highest 1 hour after radiation. So if you look at hemoglobin, we see that oxygenated hemoglobin has a very similar pattern to oxygen saturation, such that after dose 1, immediately after dose 1, the content increases but decreases again. However, we didn't see any significance over here. Given the fact that deoxygenated hemoglobin is not really changing, I am speculating if we increase the sample size, we will see significance in oxygenated hemoglobin. Then that way we can conclude that changes that we saw in vascular oxygen saturation is mainly because of oxygenated hemoglobin. So to test our method in a different cell line, we obtained a parental A549 human lung cancer, and we developed its matched radiation resistant clone. So basically, we took a subconfluent A549 cell lines and we exposed them to 2 grades of radiation, which caused some portion of the cells to die. And after 3 days, we repeated the same procedure, which caused more cell death. And we continued this process for 25 fractions. And that is how we ended up with a radiation-resistant model of this cell line. Now, when we injected these cell lines to animal model, what we saw was that in radiation-sensitive, in parental cell line, the change in oxygen saturation is significantly different 48 hours after radiation, whereas in radiation-resistant cell line, radiation-resistant tumors, this significant difference was 24 hours and 48 hours after radiation. So in summary, we validated the sensitivity of diffuse reflectance spectroscopy with an established method and proved that SO2 is correlated with hypoxic fraction, and interestingly, scarring is positively correlated with hypoxia. We also saw that radiation causes significant changes in significant increase in scarring intensity and decrease in scarring power over time. Also in lung xenografts, we observed that radiation induces reoxygenation after each dose. And in lung xenografts, we observed that reoxygenation is higher in radiation-resistant group compared to radiation-sensitive group. Now, to understand the differences that we observed among head and neck lung tumors and head and neck tumors and lung tumors, we first need to investigate reoxygenation kinetics in radiation-resistant head and neck cancer, because what I reported here was radiation-sensitive head and neck cancers. We also would like to understand why the reoxygenation kinetics are different in different tumor sites, and we have some preliminary evidence that our lab recently published that radiation-resistant lung cancer cells decrease their oxygen consumption and increase HIF1 expression. And after all, we would like to compare all of these in response to chemotherapy and combined radiation and chemotherapy. At the end, I would like to acknowledge everybody's help and support from my laboratory. And thanks to Arkansas Bioscience Institute for financial support. And thank you for your time.